Hey everybody, welcome back. Miss Calabrese here. In this video, we're going to be talking about genetic mutation, recombination, and horizontal gene transfer in bacteria. All right, so first off, a mutation is just any change in the genetic code. So any change in the sequence of DNA is considered a mutation. And mutations are the driving force of all evolution, right? It's the only way that we actually get new genetic information into our code. So mutations can happen in a few different ways, but as long as we understand that there are any kind of change in the code, that's the basis, right? So they can happen spontaneously, meaning a random mistake happened, and usually this is an error in the replication process. So remember in our replication process of DNA where we've got the unwinding with helicase, complementary base pairing with DNA polymerase III, and then the joining with ligase, sometimes there are errors where DNA polymerase III puts down the wrong nucleotide. And in that case, that would be considered a spontaneous mutation. Um, we also can have induced mutations. Induced mutations are ones that result from an exposure to something called a mutagen. So a mutagen is basically anything that's capable of mutating your DNA. And so that can be things like radiation. All right, so you spend too much time behind an X-ray machine, you spend too much time out in the sun, that's too much UV radiation that can cause mutations. Or certain chemicals can act as mutagens where they're going to disrupt your DNA. All right, so whether a mutation is spontaneous or induced, it's capable of having many different effects. Right, so some mutations are totally neutral, meaning that you can have a change in your DNA that does not result in a change in a protein that has no actual impact on the livelihood of that cell. So in that case, that would be completely neutral. That's a neutral mutation. So it doesn't really cause any adverse or beneficial effects. And on the other side of that, we can also have lethal mutations, and lethal mutations cause effects that are so harmful that it's going to lead to the death of the cell. Um, and then there are some mutations that can be beneficial. Those are rare, right? Because for the most part, organisms are pretty well adapted to their environment, but every once in a while, a mutation comes along that's beneficial. And in that case, then it would likely lead to the production of more offspring for that particular cell, and we would see that mutation take hold in the population. Okay, so we're going to mostly be talking about point mutations here. So a point mutation is when we just have one simple change in one letter of our code, right? So only a single base, one single nucleotide gets changed. And so point mutations can have several possible impacts on your cell. So a point mutation can be considered a missense mutation. A missense mutation is where we've got a change in the code that's going to lead to a different amino acid. So remember that each three letters in the code is going to be equal to one amino acid. So if we have a different letter that gives us a different codon and gives us a different amino acid, then that's considered a missense mutation. Again, missense mutations, sometimes the protein that they make can still work just fine, but other times that protein doesn't fold correctly, and then it becomes non-functional or less functional than it would have been. Right? So another type of mutation that we can get from a point change is a nonsense mutation. So nonsense means instead of having a normal codon that codes for a normal amino acid, we change it into a stop codon. And a stop codon is going to prematurely shut down translation of this protein, which means our protein is going to be much shorter than it normally would have been, which is usually going to make it non-functional. Um, and then it's also possible to have silent mutations. So silent mutations give us a different codon, but that different codon doesn't necessarily cause any problems because it codes for the same amino acid. So we can still kind of consider that a neutral mutation because it's not causing any problems. So let's look at this image over here for kind of an example. So if we're looking at the code in our DNA, we'll start right here and look at, say we have a three-letter sequence TTC, so thymine, thymine, cytosine. When we go to do transcription and make our mRNA, we're going to transcribe that TTC into an AAG, right? Because T becomes A, T becomes A, and C becomes G, right? And if we look at AAG in our genetic code, we can see that the codon AAG always codes for the amino acid lysine, right? And here's what the lysine looks like down here. Right, so TTC becomes AAG in the RNA and the protein is lysine. Now, say we have a mutation there. So here we can see what a silent mutation would look like. So instead of TTC, we had an error, and now we've got TTT. So our mRNA is now going to look like AAA, and that, guess what, is still going to give us lysine. So even though there was a mutation there, that mutation was silent, so we still ended up with the same amino acid, which means our protein is going to fold just exactly the same, and we have no problems here. Say, instead of TTC, we get TCC. So that's going to give us AGG as our codon, which gives us a different amino acid. In this case, it's arginine. So arginine, being a different amino acid than lysine, means that this is now a missense mutation. So missense gives us the wrong amino acid. Right? If we change TTC to ATC, now our codon is UAG, and that's a stop codon. So that means we're not going to put down an amino acid at all. We're just going to chop the protein short at that point. So that nonsense mutation would be pretty dramatic. Right? Another type of mutation that we can have, and this one, instead of just changing a letter, for these frame shift mutations, we're either inserting a letter or deleting a letter. So instead of just changing one around, we're adding one or taking one away. So if we look at our normal sequence here, um, where we can see the letter codes, if we are adding in a letter where there shouldn't be one, we can get a different, not only do we get a different amino acid for that particular codon, but now we're kind of pushing the whole code down one letter at a time. And because we're pushing it down one letter at a time, we're shifting our reading frame. And that's going to give us a different amino acid all the way down. So that's a frame shift mutation. And those are almost always non-functional proteins because it's basically nonsense, right? We've changed every single amino acid, so it doesn't even resemble the original protein that was coded for in the recipe at all, right? So frame shift mutations are almost always the most devastating type of mutation that can happen. Right, so how do we fix this? So you do have some built-in strategies to repair mutations. So your DNA actually proofreads itself. So we mentioned that DNA polymerase 3 is the one that's responsible for complementary base pairing during replication. But you also have some other DNA polymerases that are really good at reading that DNA and double checking to make sure there are no errors. Right, so if there is errors, then we have to go and fix those errors. So normally light damage can be repaired easily. So for example, a little bit of UV damage, you spend a little bit too much time in the sun, your built-in repair mechanism can go fix whatever problems happened in the DNA and you'll be just fine. However, if you get too high exposure to mutagens like UV light, then we can cause widespread damage that your repair mechanisms just aren't able to keep up with, and that can lead to things like cancer. Right, so the main process here for repair of mutations is called excision repair. And excision repair means we're just reading that DNA strand, and when we see an error, we're going to use an enzyme to cut out the bad letter and put in the good letter. So here's what that looks like. So we've got, so if this is our one side of the DNA strand and this is the other side, we can see that these particular base pairs over here don't match with these ones over here, right? So if they don't fit, then we need to get our enzyme in place to cut those bad ones out, replace them with good ones so that they fit with the original DNA strand, right? So that's excision repair. We just use an enzyme to cut out the bad letters and add good letters back in, right? And so mutations, because they happen in the DNA, if they happen, especially for humans and things like us, if they happen in the sex cells, in the sperm and egg, they can be heritable. If they're happening, if we're talking about bacteria, any genetic mutation could be heritable, meaning they can pass it on to their offspring. Um, Again, most mutations are not going to be beneficial because we kind of assume that the organism is working at its most efficient level. But every once in a while, a beneficial mutation comes along. And when it does come along, it takes hold in the population. Um, and mutations are mostly useful for changes in the environment. So if the environment of a critter changes, then we might need a mutation to deal with those changes and be most well adapted for our new environment. All right, so which of the following types of mutations has the most devastating effect on a cell? And the answer is B, a frame shift mutation is going to be the most devastating because usually it will affect every single amino acid from the point of the shifted frame on down. All right, so let's switch gears here a little bit and talk about DNA recombination. So recombination is anytime we share DNA. Bacteria we don't think about them recombining in the same way that, say, humans recombine, right? We do genetic recombination every time we mate and produce offspring because we're shuffling the genes and creating a totally new individual. Bacteria don't reproduce that way. They reproduce through binary fission, which is essentially they make an identical copy of themselves. So there's no DNA shuffling that happens there. So recombination means we have to do something a little bit different in bacteria. Right, so we get this done by horizontal gene transfer. So in humans, we transfer genes vertically from parent to offspring. In bacteria, they can transfer genes vertically from parent to offspring, but they can also transfer them horizontally, meaning they can transfer from adult bacterium to adult bacterium, from one organism to another directly. And there are a few different ways that this can happen. One of the most important ways that horizontal gene transfer is possible in bacteria is through structures called plasmids. Right, so we mentioned plasmids earlier in the course, how they were usually small circular pieces of DNA that contain one or a few genes on them, so not many genes. It's separate from the central chromosome of that particular bacteria. And these plasmids are able to be copied and transferred to other bacteria. All right, so three main types of horizontal gene transfer, and those are conjugation, transformation, and transduction. So conjugation requires a direct connection between two bacterial cells. So this means that one bacterial cell that has a plasmid is capable of building a pilus, which is a connecting structure like a tunnel, from that bacterial cell to a neighboring cell and send a copy of his plasmid over to his neighbor. So it's a direct transfer through conjugation. Transformation involves bringing on DNA from the environment. So just naked DNA that's floating around in the environment, able to be absorbed by certain bacterial cells. And then the last type here, transduction, is when we're moving DNA from one bacteria to another based on a virus. So a virus or a bacteriophage is the one that's actually transferring that DNA from one cell to the other. Again, these are all types of horizontal gene transfer where bacteria are mixing up their DNA. All right, so of those three types, the only type that's direct is conjugation. So this conjugation is a direct interaction between two cells that are alive, where one cell is actively transmitting genes to another cell. Transformation is indirect because usually the donor DNA, the DNA that came from whatever donor cell, is dead, and that's why it's naked DNA floating around in the environment. So we need a live cell to take up that floating DNA from the dead donor. And then in transduction, usually the donor is a bacterial cell that's been lysed, essentially, lytic cycle of a viral infection, and then that virus takes that DNA and moves it to a new bacterium. Okay. So in terms of conjugation, we'll get into this one a little bit more specifically. Both gram-negative and gram-positive bacterial cells are capable of conjugation. We're going to focus mostly on what gram-negative conjugation looks like. It basically looks like a plasmid, which is inside that gram-negative bacterial cell. On that plasmid are genes for how to create a pilus. So pilus is just this tunnel-like structure that can connect to bacterial cells. And the genes for that pilus are on the plasmid, and that we call that an F factor, a fertility factor, meaning that it's got the ability to create this pilus and reach out and grab another cell. So the cell that has the fertility factor is considered F positive or fertile, and the cell that doesn't, its neighboring cell, is going to be F negative, so he's not going to have that plasmid until he's contacted by the F positive cell, right? In gram-positive conjugation, it's a little bit different, where we're creating an opening between two adjacent cells instead of building this pilus. So let's look at, I think I have a picture here coming up of what this looks like. Um, Sometimes these plasmids that are transmitted from one bacterium to another contain genes for resistance. And resistance means referring to the ability of bacteria to be resistant to antibiotics. So if a plasmid contains genes that contribute to antibiotic resistance, we call it an R factor or resistance plasmid. Okay, and so here's what this looks like. So here we've got our donor cell. So this is an F positive cell here. And we can see inside this cell we've got the regular chromosome. And then separate from that regular chromosome, we've got a plasmid. The plasmid is going to have genes on it that will allow this donor cell to build a pilus, and that pilus is this bridge that connects these two cells. So he builds a pilus, reaches out to this neighboring cell who's F negative, and what he's going to do is make a copy of his plasmid and send that copy of the plasmid over to that F negative neighboring cell, right? So now, not only is the initial guy fertile or F positive, now the neighbor who was once F negative is now fertile as well because he contains the plasmid, and that plasmid has genes on it for how to build a pilus. So you can imagine in this situation, if we've got, say, genes on this plasmid that are making these particular bacteria capable of resisting antibiotics, that this can spread very rapidly in a population because now this recipient cell is also fertile and he can go spread these resistance genes to other cells, and then they can spread it to other cells and they can spread it to other cells, and pretty soon those antibiotic resistance genes have spread all throughout this bacterial population. All right, so the next type of horizontal gene transfer is transformation. And this one's a little bit different, so this is not a direct process. It involves having a competent cell. So a competent cell is one that's going to be capable of bringing in genetic material. So he's got DNA binding proteins on the surface of the cell. So he's got these proteins on the outside surface that he can use to bring in naked DNA from the environment. So DNA that's just floating around. If he's got DNA binding proteins, then he's considered competent and he can bring those DNA molecules inside, and that's the process of transformation. Right, so we don't need anything except those DNA binding proteins on the surface of the cell. And this is super useful because we can do this kind of thing in a test tube, right? If we wanted to do genetic recombination and add some genes to a particular bacteria, then all we need is competent bacteria, and we can add the genes to the test tube, and those competent bacteria will uptake those genes and incorporate them. All right, and then the last type is transduction, and transduction I mentioned is when we use a virus or a bacteriophage to move DNA from one bacteria to another. So let's look at the pictures here. So in this conjugation, we can see the plasmid is helping us create the pilus, and the pilus is how we're going to transfer that plasmid from the donor cell to the recipient cell. In transduction, you can see the phage on the outside of here, that virus, who's infecting this cell. When he infects this cell and lyses it, some of the new baby virions, those little tiny viruses that are created from the lytic process, will be carrying some of the DNA from the bacterial cell, and they can then transmit it to other bacterial cells. So that's transduction. And then transformation, again, is just uptake of naked DNA. So this particular bacteria here is going to have DNA binding proteins on his surface that's going to allow him to pick up these plasmids that are just floating around in the environment, bring it into the cell. Right, so those are the three main ways of horizontal gene transfer in bacteria. Okay, one other quick note as far as genetic things that can happen inside these cells. Transposons are jumping genes, so transposable elements, meaning these are genes that can move from one part of the genome to another. So, and they can also move from the genome, so the main chromosome of the bacteria, to a plasmid. So even genes that are not on a plasmid can possibly move to a plasmid, and that can make them much more transmissible because now they won't be transmissible just to the descendants of that particular bacteria, but also they're going to be capable of horizontal transfer as well. All right. One question here. So which of the following mechanisms of horizontal transfer involves the transfer of bacterial DNA through a bacteriophage? So where is bacteriophages come in terms of horizontal gene transfer? And the answer is transduction. So transduction is going to be the transfer of DNA through a bacteriophage. So I hope this was helpful. Let me know if you have any questions.